Hello, my name is Aaliyah Calderon, and I will be doing a speech on cloning today. I am trying really hard to get this video up, so unfortunately I do not have an audience, but I will be using my webcam as my audience today. So, in 1996, Dolly was the first sheep to be cloned out of 277 attempts. She was created by using the nucleus of an udder cell from a 6-year-old white sheep named Finn Dorset. They reprogrammed the cell and injected the cell into an unfertilized egg from a black sheep, which had its nucleus removed. The scientists cultured the egg for about 6 days for it to develop into an embryo and then implanted it into a surrogate mother. Unfortunately, due to the way they cloned her, her nucleus was already aged by about 6 years, so she only lived from July 5th, 1996 to February 14th, 2003 because scientists had to euthanize her. She suffered from arthritis due to the aged nucleus used in cloning. Sheep lived for about 11 years, but Dolly lived until she was 2 years and 10 months. The many scientific innovations that resulted from cloning include cloning in medicine, reviving extinct and endangered species, cloning a deceased pet, cloning for drug production, and cloning stem cells for human research. Cloning in medicine, cloning animal models of disease by using cloned mice that are genetically engineered to carry disease-causing mutations in their genes. Cloning transgenic animals can shrink the time they could take to naturally reproduce these types of animals for medical research. So they would use white lab mice that were genetically engineered to carry a disease that would be incurable at the moment, but then they use them as research to find a cure. They could also clone to make stem cells. Stem cells build, maintain, and repair the body throughout our lives because these are processes that stem cells do naturally. They can be manipulated to repair damaged or diseased organs and tissues. Transferring stem cells can be seen to the body as foreign and trigger an immune response. So when people do stem cell transplants, it can be hard sometimes because the stem cells will be rejected from the body, even if it's a match. Research is looking into cloning stem cells to be identical to a specific individual's stem cells to avoid rejection. In 2013, scientists at Oregon Health and Science University were the first to use cloning techniques to successfully create human embryonic stem cells for an 8-month-old with a rare genetic disease. Reviving endangered or extinct species, the first thing you would think of is Jurassic Park. Most people believe in theory that scientists just need a well-preserved source of DNA from an extinct dinosaur and a closely related species that is currently living to serve as an egg donor and a surrogate mother. That is not the case. In 2009, scientists had their first near success with resurrecting an extinct animal. They used goats as egg donors and surrogates to make several clones of a wild mountain goat called the Bucardo. The longest living clone of this extinct species died at birth. Cloning endangered animals is much easier for scientists to do, and wild species have already been cloned multiple times. Cloning endangered animals is easier because they can donate healthy living cells, and animals such as cattle, deer, bison, coyotes have been cloned. Genetic diversity can become an issue for endangered species, and cloning does not help change that. So there is a reason why animals are endangered. Just because you clone more to help them doesn't mean you fix the problem environmentally. Reproducing a deceased pet. Now you can clone your pets, cats or dogs. On December 22nd, 2001, a kitten named Cece was the first cat and the first domestic pet to be cloned. The original pet was named Rainbow, and she was cloned to create an identical cat to herself named Cece. Genetically, they are the same, but identically, they do not look alike. I do not have any pictures, but they do not look exactly alike, but genetically, they are identical. In South Korea, scientists have also cloned 4 beagles with glow-in-the-dark skin. 4 puppies named Ruby and Puppy combined. These puppies glow red under ultraviolet light and look normal in the daylight. That's its normal, and that's its glow in the dark. The dog's nails and abdomens look red to the naked eye. This occurred in 2009. Also, drug production. Farm animals such as cows and sheep are being cloned in order to produce drugs and proteins that are used for medicinal purposes. For example, scientists use a cow that is known for producing a high quantity of milk. Then the scientists can take living cells from that cow to genetically engineer it to carry the codes for a specific drug or vaccine. Then they could reproduce the clone of the original cow that can now produce milk with drugs in its milk. And since the clone carries a gene for the drug, it can pass it to the offspring and they can produce milk with drugs. So cloning humans. The idea of cloning a human is highly controversial. Lawmakers and scientists see cloning in order to create a human baby immoral. Scientists see it as a possible solution for couples with infertility issues, and even though this process of cloning has had its successes, the process is still difficult to perfect and is inefficient. The success rate for cloning is quite low because most embryos fail to develop and successful embryos can miscarry. Current human cloning is used to recreate embryonic stem cells for medicine and research, as discussed earlier. So even though cloning can be a great scientific venture, there can be downfalls. So with everything scientific, there's always good things and bad things. That is the end of my speech. I hope you enjoyed, and thank you very much.